buenos días tengan todos ustedes. Soy el doctor Oscar Valencia, coordinador de este espacio de educación médica continua. Y antes de dar inicio a nuestra sesión, quiero recordarles que hay que mantener en silencio nuestros micrófonos. Todas las preguntas las podrán realizar a través del chat y al final de la sesión se las haremos llegar a nuestra ponente. También es importante que nos hagan favor de llenar una encuesta de satisfacción al final de la sesión y recuerden poner su nombre completo en la pantalla con el fin de poder tener el nombre para las constancias. En nombre de la Escuela de Medicina y Ciencias de la Salud, les doy la más cordial bienvenida a esta sesión académica de pediatría. On behalf of the School of Medicine and Health Science of Tecnológico de Monterrey, we'll welcome you to our session, to our grand rounds. It is really a privilege to have Dr. Laura Thornton with us. I'm going to give the floor to Dr. Eva Trujillo. She's going to be the one who's introducing our speaker for today. Adelante.
Gracias, Oscar. Laura, thank you for being here. We're very pleased to have you here. Voy a hacer una introducción corta de Laura. ¿Empiezas la proyección, Oscar? La doctora Laura Thornton es profesora en el Departamento de Psiquiatría de la Universidad de Carolina del Norte en Chapel Hill y ella es la subdirectora de la Iniciativa Internacional de Genética de Trastornos de la Alimentación conocida como EGGI. Se ha desempeñado como coordinador internacional de investigación de trastornos de la conducta alimentaria durante 20 años. Tiene una amplia experiencia en el diseño de instrumentos, la recopilación, la gestión de datos, el análisis y la difusión de los resultados de la investigación epidemiológica comunitaria a gran escala. Su enfoque en la investigación incluye la evaluación de los factores genéticos que influyen en la predisposición y el estudio de los aspectos fenotípicos de los trastornos de la conducta alimentaria y la obesidad, ya que la identificación y comprensión de los factores de riesgo de trastornos alimentarios y las conductas alimentarias de riesgo ayudará en los esfuerzos de prevención, detección, tratamiento y eventualmente mejorará los efectos de estas enfermedades devastadoras. Ella está comprometida con el avance de nuestra comprensión de todos los aspectos de los trastornos de la conducta alimentaria, tanto desde una perspectiva etiológica como del desarrollo. So, sin más preámbulos, Laura, it's an honor for me to introduce you to our community, to our medical community in the Tecnológico de Monterrey. So welcome and please start.
Thank you. Thank you for having me today. I'm going to share my screen so we can, let's do that, share and, oh. Oh, let's do it then. Sorry.
Now, yes, now it's ready.
Okay, thank you. Thank you. All right. So thank you for having me here today. First off, I want to send greetings for Dr. Cynthia Bulick. She and Dr. Trujillo have been working closely together to get EGGI up and running in Mexico, and we're going to get to that a little bit later. But she is the leader of the research efforts that I'm going to be discussing today. She's the worldwide leader, and she has graciously provided me with the slides for today's presentation. So with that, I'm going to get started. Now, my goal today is to provide you with a summary of recent research findings related to the genetics of eating disorders. We're going to start with some pretty basic stuff just to make sure we're all on the same page. Then we're going to briefly discuss translation to patients and advocates. And lastly, we'll finish with what is happening in the genetics research world now. All right, so very briefly, I'm sure you're all aware, there are several different eating disorders. These eating disorders have some overlapping symptomatology, but they each have unique features. So for anorexia nervosa, it's characterized by low weight, an intense fear of weight gain, an inability to recognize the seriousness of the low weight. Bulimia nervosa and binge eating disorder share the core feature of binge eating. They can occur in people of normal, overweight, or obese body sizes. The difference between them is compensatory behaviors. Binge eating occurs with compensatory behaviors in bulimia nervosa and is distinctly absent in binge eating disorder. And the latest eating disorder to join DSM is avoidant restrictive food intake disorder. And with this disorder, there can be nutritional deficiencies, there can be weight loss or failure to gain. And of course, presentation and course of illness can be complicated. So over time, patients may transition from one disorder to another. And so what we have here is, this is taken from data from Sweden. And in Sweden, individuals who go to an eating disorder clinic have their diagnoses registered in these big population registers, hospital registers, but these are particularly eating disorder registers. And so what we have here in this first column are individuals who have gone in for initial presentation to the clinics, and these are their eating disorders. The size of the circle indicates how many people, about how many people relatively, have each of the disorders. So the majority of people that go into the clinic have eating disorder not otherwise specified. A little fewer have anorexia nervosa, and then binge eating disorder is of course the smallest population. The second column of circles is what we see 1 year after initial, so 1 year follow-up, and the third column is two-year follow-up. The lines between the circles indicate how patients move from one disorder to another between time points. So in this case, and the thickness of the line indicates how many people are essentially going from one disorder to another. So the majority of people who start out with anorexia stay with anorexia at time 1 and then at time 2, the follow-ups. However, there are some that go into remission, some that go to EDNOS, fewer that transition to bulimia, and even fewer, although you can't really see the line, go to binge eating disorder. So yes, people can move between one disorder and another, which will complicate things genetically a little bit later. But what do we know about the genetics of the epidemiology of eating disorders? Well, we do know that between 1% and 4% of the population will have a lived experience with the eating disorder. And although all three have increased standardized mortality ratios, the mortality for those with anorexia nervosa is particularly high. In addition, twin-based heritability estimates are between 45 and 60%. So the take-home message here is that eating disorders are serious illnesses, and a substantial proportion of the liability to them is due to genetic factors. So risk, there's a great deal of genetic factors in risk for eating disorders. Now we're going to because our genetic analyses currently involve mostly anorexia, we're going to dive down a little deeper into anorexia. So it's particularly interesting for a number of reasons. First, starvation is reinforcing, whereas for most of us, being hungry is uncomfortable. Second, fats are aversive. Those who with anorexia report not liking the smell, the taste, or the mouthfeel. Most of us find that fat adds a richness to foods and enhances texture. Activity is more reinforcing than food. So people with anorexia will tend to want to move rather than eat. And in fact, we now have animal models of this particular trait of anorexia. It can also be very difficult to renourish patients. They often enter a hypermetabolic period during renourishment, where it is hard to determine how many calories they need to gain weight because their bodies burn calories so fast. And people with anorexia tend to lose weight after renourishment because their bodies revert to a negative settling point. So what does that mean? Well, most of us find that starvation or a negative energy balance where we expend more energy than we actually consume, make us irritable, anxious, or tired, and may make us engage in food-searching behaviors. However, those with anorexia state that starvation actually creates a sense of calm, and in fact can be anxiolytic. And doing genetic research about anorexia does not just tell us about anorexia. It's going to inform us about traits such as nutrition, metabolism, and physical activity, which is why I focused on those particular features in the last slide. And it may help us understand obesity. Genetic research can also provide new information on commonly comorbid disorders like major depression and anxiety. So there's a wealth of information that can be obtained from studying eating disorders that goes beyond just eating disorders. So with that overview, we're going to dive into genetics. The primary analytic method right now to study the genetics of disease is a genome-wide association study. So we're going to go through some of the basics. A genome-wide association study, or GWAS, is an approach used to associate specific genetic variations with particular diseases. The method involves genotyping a large number of cases, and for us, this might be individuals with anorexia. These people can have a lifetime history of anorexia, so have had it in the past, or can be currently experiencing symptoms. The status of illness doesn't matter because your genes don't change. We also need DNA from a large number of people who we consider controls, and these people have no history of any eating disorder in their past. Then we look at markers across the genome. So here's our 23 chromosomes. Each chromosome is taken, and several different areas, which we'll call loci or SNPs, are within each chromosome. And we're going to look at these across every single person. We are literally looking at 1 million different places for genetic markers that can be used to predict the presence of the disease. Then we compare all of the cases to all of the controls to see where the differences lie. And here you can see we have many places where the cases and controls are the same, but in this box, the cases and controls are different. This is what we're going to call as variant 1. For our example, it is less common for controls to have the C version of this SNP or area, and more common for controls to have the T. And we can see that in general, 62% of cases right here have the C, where only 30% of controls have the C variant. We then go through and we do this for every single variant or area in our genome. So 1 million different times. So here for variant 2, we looked at the differences and we find that they're very, very similar. And we conduct regression analyses to determine whether these differences are significant. So in this case, it's a significant difference. In this case, it is not. When we've done this and we've gone through every single variant, so 1 million different times, we get something called a Manhattan plot. And it's called a Manhattan plot because it looks like the skyline of Manhattan. So I'm going to orient you to this graph. Along the x-axis here are the chromosomes. So you can see 1, 2, 3, all the way down to 22. And here we've included the X chromosome. The size of the on the axis for each chromosome is about the relative size difference for each of them. So chromosome 1 tends to be the biggest. It occupies the largest area on the x-axis. 22 is the smallest. It occupies the smallest. The y-axis indicates the significance level, right? And this red line that we've put all the way across is the critical significance level. Anything above that line is considered to be significant. Now, you will note that our p-value is set to 5 × 10⁻⁸, which is way larger than any p-value we do in other research. But we're doing this because we are doing over 1 million tests. So we need to control for multiple comparisons. And setting a stricter p-value helps us reduce the number of false positives. We do this because we want to know that the loci or areas that we're going to focus on are actually worth trying to figure out. Now, all the SNPs or areas or variants, whatever you want to call them, are points in this graph. So that would be 1 million different points that we have represented here. The Manhattan plot in this particular Manhattan plot has several, as you can see, several areas or locations or SNPs that are significant, that are significantly associated with the disease in question, right? And those are indicated by the diamonds. This is a really nice looking Manhattan plot, right? It actually does have several different areas. Now, so where are we with eating disorders? Well, we knew ahead of time that sample size was going to be the most important factor for GWAS. So to achieve large samples, collaborations are essential. Researchers from many institutions began working together to study the genetics of eating disorders through the Psychiatric Genomics Consortium, or the PGC. The central idea of the PGC is to leverage global collaboration to advance genetic discovery of biologically, clinically, and therapeutically meaningful insights. The eating disorders of the working group of the PGC was formed, and our group now has members from over 20 countries around the world. But I'm getting a little bit ahead of myself. So in the early days, when this group came together, we completed a GWAS for anorexia nervosa using a few thousand cases, which we thought was fantastic. We combed research institutes and clinics, and most of our participants, or most of the samples that we got, were from patients. So people who were actually seen in the clinic, were clinically diagnosed, and we actually had blood samples for. We found only one significant SNP. So our Manhattan plot looked more like a village with a bell tower and not the skyline of New York. And it became apparent that we needed much larger sample sizes. So Dr. Bulick said, okay, well, how are we going to do this? And she got a huge grant from the Klarman Family Foundation to initiate the Anorexia Nervosa Genetics Initiative, or ANGI. And so at this time, this was the largest study of eating disorders ever taken on. 4 sites were involved in the data collection: the University of North Carolina at Chapel Hill in the United States, Karolinska Institutet in Stockholm, Sweden, QIMR Berghofer in Australia with the help of the University of Otago in New Zealand, and Aarhus University in Denmark. Now, we changed things up. As you know, to go through assessments in person with patients takes a lot of time. So the very first thing we did is said, okay, how can we streamline this? How can we actually get this type of information? And we developed an online survey that's based on the SCID, so based on an actual interview, to determine case control status. So people would go in, we would consent them, they would go in, do this particular survey, and it would tell us whether or not they met criteria for case or control status. If they met criteria, we then sent them kits to get their blood drawn, so we could actually get DNA from their blood samples. And doing this, just these few changes, which is the online screening, in just 3 years, we were able to collect samples from 13,000 participants with a history of anorexia and 9,000 controls, which was amazing. In order to do this, we also activated the eating disorders community, and we were some of the earliest researchers to use social media for recruitment. Well, with our lesson learned from the first GWAS about sample size, we put the data together from Angie. So here's all of the data from Angie with the data from the first GWAS, which is this here. And then we combed the world for other samples, and the UK Biobank and the Wellcome Trust Case Control Consortium added to our numbers. In total, we ended up with data from nearly 17,000 cases and over 55,000 controls across 17 countries. The samples from Angie went a long way to boosting our numbers, as you can see. So what did we find? Well, this is the Manhattan plot of our anorexia GWAS. And remember, we're looking at SNPs or loci or variants that are above the red line, because these indicate that they significantly increase the risk of anorexia. And we have 8 significant hits. So there's all these little green diamonds. 8 distinct areas in the genome were found to be associated with anorexia, which again is, I think, amazing. When we reviewed the literature, we found that these SNPs were also associated with other features, with some autoimmune illnesses, some metabolic traits, neuropsychiatric disorders, and sex hormones. And, as I said, I think this is great, but we can get even more from this graph than what we're showing right here, because there is valuable information that's actually below the red line. And what do I mean by that? Well, taking the information we get from GWAS, so all of it, not just the significant SNPs, we can estimate genetic correlations between traits from published summary statistics. And what does that mean? Well, in the past, we would have had to have the same measures on the same people to get correlations, right? You'd have to measure depression, anxiety, eating disorders, all on the same individuals. The methods we have today to estimate genetic correlations don't require the traits of interest to be measured on the same people. We can now estimate a genetic correlation, for example, between anorexia and anxiety by having GWAS results from a study, say, in the United Kingdom, studying anxiety, and another large sample from a study in Sweden on anorexia. So between different diseases, it's the genetic analog of comorbidity. Now, it's important to note that these differ from phenotypic correlations. Genetic correlations effectively describe the genetic architecture of complex traits and diseases. They estimate the extent to which the same genes are operating across different traits. Like phenotypic correlations, they can be positive, indicating that the same genes are working in the same direction, which means that they are increasing the risk for both. Or they can be negative, indicating that the same genes are working in opposite directions, where they're increasing the risk for one trait and decreasing the risk for another. Now, I don't want you to try and read all of this. We're going to walk through it, and then I'm going to break it down in the next few slides. But this shows all of the significant genetic correlations of anorexia that we found. We looked at hundreds of traits. These are just the significant ones. So the traits are listed down the y-axis. The x-axis indicates the correlations. The red dotted line in the center is a correlation of 0, which means there's no association. Everything to the right of the line is a positive correlation. Everything to the left of the line is a negative correlation. All right, so let's take these apart and look at them individually here. So we're starting at that top of the previous graph, and we can see that there is nothing on the negative side. All the correlations for this piece are positive. There are several strong correlations with various psychiatric traits. The strongest is with OCD. You can see that right here. This means that many of the same genetic factors that influence OCD also influence anorexia. The other positively correlated traits are depression, schizophrenia, anxiety, and neuroticism. So what we're seeing at the genetic level complements what we see in terms of comorbidity within the patient population. Next, we see correlations with several measures of education. At some level, whatever the drive is to succeed might be correlated with both education and anorexia. We also found a correlation with physical activity. This is objectively measured physical activity. So it's not just people reporting, oh yeah, I run 3 miles a day or whatever it is. This might indicate that that what drives someone to move or be physically active or exercise might share genetic factors with those with anorexia. All right, the next group are metabolic factors. And we see our first negative correlations here. And the negative correlations are with insulin, with leptin, and with type 2 diabetes. And these tend to be considered unfavorable metabolic parameters. On the other hand, HDL, or high-density lipoprotein, is a favorable metabolic parameter, and it is positively genetically correlated with anorexia. I should note that other psychiatric traits do not have such a strong association with the metabolic traits. So this is evidence that perhaps anorexia is also a metabolic disorder. All right, at the bottom of the graph, of the first graph, we observed negative genetic correlations with anorexia with several anthropometric traits. So you can see her body fat percentage, BMI, obesity measures. So what does this mean? Well, some of the same genetic factors that influence obesity and body weight are also influencing anorexia, but in the opposite direction. So in review, we found that anorexia is strongly positively correlated with psychiatric traits, with education, and with physical activity. It is negatively correlated with unfavorable metabolic traits, but positively correlated with favorable metabolic traits. And anorexia is negatively genetically correlated with body measurement variables. And this picture is going to help us change the way we think about anorexia. Anorexia is both a psychiatric and a metabolic disorder. Okay, so what are the take-home message? We need to reconceptualize anorexia as a metabopsychiatric disorder for a number of reasons. First, patients with anorexia have an astounding ability to reach and maintain a low weight. Second, they often return to a negative settling point after renourishment. Next, we see negative genetic correlations with BMI and other unfavorable metabolic parameters. We also see a positive genetic correlation with HDL. And last, patients with anorexia feel better when hungry than when sated or fed. Okay, so now we're going to move on to how we integrate this into personal narratives. So what does this mean for your patients? Well, the first thing that has to happen is translation of research results. This particular graphic here is a very sad one, and it's certainly an example of science gone wrong and not put into context. The truth, bleach does kill germs, but it should never be ingested or used on one's body, as was suggested for the coronavirus at one point. So it's the researcher's responsibility to interpret the findings. We also have to provide context for them. Are the findings applicable to populations at large, or can they be applicable to the individual? Are there clinical implications? Then it's the clinician's responsibility to have a general understanding of the research and to be able to assist patients and families. With this, genetic counseling in particular can help patients and families understand the research and how it applies to them. And I want to emphasize this because genetic research is changing so quickly these days that it's hard to keep up with all the new things coming out. Okay, so what do we share with patients and families? First and foremost, genetics is just one piece of the risk puzzle. Environment plays an important role. Timing of events can be important as well. And then there is the interaction of all of these factors. Risk is not all or nothing, not genes or environment. It is both nature and nurture when it comes to eating disorders. There are challenges to understanding probabilities. Calculating the probability that a particular person will get an eating disorder is nearly impossible. We can say things like, we know that if you have a family member with a history of eating disorders, your risk is increased above the general population. We have to also indicate that probability is not certainty. When it comes to genetics, this means that genetics for eating disorders does not mean destiny. Genetic guilt. This is a hard one. It's now fact that eating disorders have a genetic component. This means that parents may feel guilt over their child's illness because they are the source of their child's genetics. But here's the important point. We cannot change the genetics of our children. We have to remember that the child's genetic makeup is only one factor contributing to the disorder. The genetic makeup of the child also contributes to many wonderful things about the child. We need to emphasize that eating disorders are genetic and not psychological. We have to avoid oversimplification. Eating disorders are complex illnesses with multiple factors, including multiple genetic factors. You can't say, I have the gene for eating disorders. And in the end, there is no genetic testing that can be done for eating disorders. Both genes, many genes, and environment and their interaction contribute to eating disorders. So this is perhaps one model that might help people understand risk, right? Everyone's dealt a hand of cards in life. You have genetic risk, you have genetic protective factors. You have environmental risk factors, you have environmental protective factors. Everyone gets some combination of these, high or low, for whatever you're looking at. And then there's the wild card. Events, timing, interactions that are just unknown. So this is why doing risk prediction is very, very difficult for a particular individual. So now that I've emphasized all the things we need to be mindful of when speaking with patients, what is the role of genetic counseling? Well, genetic counseling can alleviate the stigma and shame patients and parents feel. It can correct misconceptions about eating disorders. It can prepare family members to intervene. It can also promote health-seeking behaviors. And all of this can potentially facilitate earlier recovery. There's been one study looking at the potential impact of genetic counseling on perceptions of causes of eating disorders. And they looked at 107 individuals with a personal history of eating disorders. And they gave them vignettes to read about genetic counseling. And what the researchers found is that individuals with eating disorders overestimated the risk for eating disorders in their children. So that this is the actual empirical data, risk for getting an eating disorder if you have a parent with an eating disorder is 7.5%, and they are 4 and 5 times, well, actually more than that, 8 times more likely to say that their child is more likely to get an eating disorder. And interestingly, every single participant overestimated their risk to daughters. So there was definitely a sex bias. Imagined counseling also, so this is, remember, they just read these vignettes. They didn't go through the counseling. So imagined counseling reduced all the negative feelings about eating disorders, including the stigma, the shame, and the guilt. And many felt that genetic counseling would be useful, as illustrated by this quote. Wow, just reading this validated so many things I felt. Like that it is genes and environment, and that guilt is so common in people with EDs. I feel like people need to hear and understand this, people with EDs and the public. All right, so how does genetic research help now? Well, we know that anorexia is a metabopsychiatric disorder. This is the new thing. Genetics provides a way to explain the illness to patients and families. And we know now that we need to pay attention to metabolic factors, as these may be very important in facilitating recovery. Genetics has also given us an explanation on why adequate nutrition is essential, and also why many patients fail to maintain their weight after renourishment. So it's not that they're just going away from their treatment. How can it help in the future? Well, we're just beginning to unlock all of the information in our genetic research. We're currently working on genetic risk scores and hope that we might be able to identify subtypes based on these. Can we find a metabolic form of anorexia? Is it different than a psychiatric form of anorexia? In the future, we might be able to predict likely course of illness and tailor treatment accordingly, which moves us closer to personalized medicine. And then we will eventually be able to understand the underlying biology, which will then allow us to develop medications based on the biology of the illness, again, contributing to personalized medicine. And in the end, we of course hope that we can eliminate mortality from these diseases. So your take-home messages are, it's our responsibility not just to do the science, but to package the information for patients and families. Clinicians should make an effort to integrate genetics into their conversations with their patients. Make use of your resources. If you work with genetic counselors, great. If you don't, create a library of resources for your patients. And lastly, help educate. There's a lot of misinformation out there, and we need to help others unlearn incorrect perceptions. So what's happening now? And this is where we are, like, in the middle of. We've embarked on another worldwide effort to study the genetics of eating disorders. However, this time we are expanding, and we're including bulimia and binge eating disorder. And we're actively working on securing funding to expand ARFID as well, although we're not quite there yet. So what's new? Okay, EDGI is not Angie. Angie was our initial foray into this area. With EDGI, our goal globally is to enroll 100,000 individuals with a history of eating disorders and get DNA from every one of them. The National Institute of Mental Health in the United States has funded collection of samples and data in 4 countries: the United States, New Zealand, Australia, and Denmark. The United Kingdom and Sweden have obtained their own funding and have launched their own enrollment. And in the near future, we hope to add Mexico. I think Mexico is very, very close. We've been working really hard with them to get their consents and all the questionnaires online for them. And I know Eva and her team have been tirelessly moving forward with this. I'm hoping it will come online in the next month, and certainly anything people can do to help recruit would be great. We also have the Netherlands almost on board. They're just trying to get their final ethics approvals. People from Puerto Rico can enroll going through the United States, but we haven't done a formal push yet, which is what we're working on. We just obtained funding to do EDGI in Taiwan, and Italy is working on their EDGI project as well. The nice thing is we are using the same questionnaires around the world. So we've gotten together, we've talked to a lot of people, and we said, what do we want to know about these folks? Besides just their eating disorder diagnosis, there's so much more we can do with genetic information. We're also working to diversify samples, right? In the past, we focused on people of European ancestry, and now we are working very, very hard to get people of all races and ethnicities. In the United States right now, we're actually doing really well on our recruitment of the Latina population, but not so well on other minorities. We're working on it though. We've also changed how we consent people. We used to have to do consents in person. Ethics boards are now allowing digital consents. We're also doing all the questionnaires digitally as well. And we no longer need blood samples. So this was a huge barrier in the past. Now we send, at least in the United States, we send people a saliva kit, which is just a small package, to their homes. They give us a saliva sample, pack it up, and ship it back to us free of charge. It is COVID safe as well. So there's a lot less interaction, which in this time, in this world with the pandemic, has been really helpful. We've also engaged the advocacy community. We've needed help. When you're trying to reach 100,000 people around the world, we need a lot of help. We need people out there talking about it and giving us, sending people to us. Which means we're also engaging clinicians, and we engage clinicians all over the place. We've got clinical centers in the United States. We've got clinicians that are doing talks for us. It's just all over, people that are taking the flyers that we have and making them available to their patients, putting them in packets for patients. All right, so we know that increasing sample size was not enough, so we're working on that, and we want to know more about comorbidity and experiences. And as I alluded to, we are really beefing up our phenotypic assessments. So this time, we're asking a lot more about their eating disorders, the patient's eating disorders history. We're asking about what their current status is. We're asking about their quality of life due to eating disorders. We're asking about treatment history. In addition, they're giving us, we're asking patients more about comorbidity. We ask about depression, anxiety, and OCD. We're asking about their lifestyle. We have questionnaires about physical activity, about alcohol and drug use, about tobacco and even e-cigarette use. And then, of course, we have environmental things that we're asking too, about life events and trauma that they might have experienced. So having all of this information with so many people, along with the genetic data, is going to help us advance our knowledge, not just about the genetics of eating disorders, but about the environmental impacts and the interaction between genes and environment. Well, this is our webpage. On it, interested individuals can join the study, read about EGGY, find facts about eating disorders, and see what is new in research and read personal stories. And in the US, we have not only a Spanish version of the website, but all of our questionnaires are available in Spanish as well. In an effort to include people globally, accurate translations are necessary. And we're working very hard to do that across all countries. And we are thankful to everyone who has been working to put the EGGY surveys into their native languages. To get enrollment started, we've had several launches across the globe with great success. New Zealand was the first to come online just before the global lockdown due to COVID. And you can see us, there's Dr. Bulick here, and I am right there. And so this is when we were able to meet in person. And I think I got the last flight out of New Zealand right as lockdown began. So this meant that the launches in Australia and the United States were delayed, and they were delayed by several months. However, we quickly made up ground, and we are actually ahead of target now for our enrollment. We've also followed up these launches with a series of Veggie Talks. So Dr. Bulick has very candid conversations with patients, clinicians, and advocates about a variety of topics. All of these are available on YouTube if you're interested. And we maintain a presence on many social media platforms as well. So I'm really hoping that Mexico will soon join us and in the EDGI community. And as you can see, this is our team at UNC right now. And the very fine print that you have up here is the list of all the researchers and clinicians who helped us understand more about the genetics of anorexia. As you can see, it does take a world. We're adding to this daily. So please follow us on all of the social media platforms, and please let me know if you have any questions.
Thank you so much, Laura, for this thorough review. So we have several questions. Before we start, I'm going to do an invitation for everybody to our EDGI Mexico. So I'm going to do this in Spanish, Laura. Nosotros estamos a cargo de EDGI México y estamos ya en el proceso de enrolar pacientes. Entonces, si alguien tiene pacientes que necesitamos controles y necesitamos pacientes, que nos los quieran mandar exclusivamente para tomar la toma de muestras, comuníquense con nosotros a comenzar de nuevo y para que podamos empezar todo el enrolamiento de pacientes. Nuestra página de internet ligada a EDGI Internacional se lanza este mes. Entonces, esperemos que ya en las próximas semanas terminemos todo esto para que podamos empezar a hacer todo el recruitment, porque esto es muy importante, ya que nos va a permitir hacer todo el rompecabezas genético de otros trastornos de la conducta alimentaria y no nada más Angie, como presentó Laura, que es el Anorexia Nervosa Genetic Initiative. Entonces, los invitamos a eso. Y lo otro es que el Congreso Mundial de Trastornos de la Conducta Alimentaria es el próximo año, en junio del 2022, en Sintermex, por primera vez en Latinoamérica. Yo estoy a cargo del comité científico y va a estar Laura, va a estar Cindy, va a estar todos los investigadores del área de los eating disorders en Monterrey. Esperemos que nos puedan acompañar. Vamos a tener un track específico para profesionales de la salud que no se dedican a trastornos alimentarios, pero que quieren saber más de trastornos alimentarios para que puedan trabajarlo en su práctica clínica, ¿verdad? Ya con esto empezamos con las preguntas. So Laura, I have several questions for you. I'm going to start in the same order that I have them here. I have one. Is there a link that you can recommend for families to learn more about genetics and eating disorders?
Yes, it's edgi.org. That's our website. Okay. That will be about EDGI. Certainly, you can also go to NEDA, the NEDA website, which I think it's neda.org, and they have lots of information in the United States about eating disorders.
Okay, another question I have is, how can we enroll patients from Mexico to the study?
Well, I think that's where you come in, Eva.
So I just said it. Okay. So I have another question. It's in Spanish. Have you discovered a relation between, I really don't understand, no entiendo, no sé si quien hizo la pregunta, Jorge Luis, quiera hacerla en voz alta porque no entiendo las siglas. TCD no sé a qué se refiere. So let's see, he used an acronym, so I don't know what the acronym stands for, okay. So I cannot translate it for you until I know that. So let's wait to see if he tell us. Okay.
I think the way I understand it is there a relationship between the lack of growth in the first year of life with someone who might develop an eating disorder as a teenager? Does that make sense?
Yes, that does make sense. And failure to thrive, we have not really been able to investigate fully because just the way we get registered data from Sweden and Denmark, those are the places that we would be able to get that kind of information. In other countries, you know, we do retrospective questionnaires, but you don't know how biased they are. Certainly, we do see an association between kids who avoid eating for whatever reason. They have the ARFID eating disorder and anorexia later on. So it is certainly possible, although again, just because there's lack of growth early on doesn't mean that that's going to definitely put them on the path for anorexia. It absolutely can be a risk factor though, or an indication that they need to be watched.
So is that something as pediatricians that we should be documenting or as a possible risk for the future?
I think you have to take a lot of things into consideration when you do that. So do they have are there other illnesses that might be causing the failure to thrive? How are you seeing their growth curve? Are they staying on the same growth curve, or are they rapidly dropping off? Because some kids are just going to be small anyway, right? And so what does their growth curve look like? There's a lot of factors you have to take into consideration. I think it's when that all of a sudden they have a dramatic drop that's unanticipated and you can't find another reason for that. Maybe you might need to be thinking, if you've eliminated everything else, you might need to go ahead and say, yeah, let's just keep a watch. Not necessarily alert the parents, but you as a clinician know that, yeah, this might be something I have to go ahead and watch.
Thank you.
Okay, so we have more questions here. Currently, we have an epidemic of obesity in children and adolescents with a high risk for diabetes and non-alcoholic fat liver disease in the future. So how can we do a balance between the intervention in lifestyle and without generating a risk for an eating disorder? Is there a genetic profile that we should do?
Okay, this is a great question because in my other side of life, we are looking at pediatric obesity. So this is outside of UNC. I work with Virginia Commonwealth University, and we are doing this. And we're trying to make sure that we are not doing harm in trying to help children with obesity, right? So things that we've done is you target the parents and have changes in the parents, particularly for younger children, right? So you don't make the child the target of the investigation or the intervention, you make the parents the child, or the parents the target of the intervention. There really is no genetic profile you can go ahead and do at this point because there are so many genes involved, and you don't have to have all of them. You can have some weird combination of things. And we have even found that people that have a very low genetic risk for anorexia, their environmental factors can push them, in fact, to develop anorexia. And those people that have many genes for anorexia or have a high genetic risk for anorexia never develop. So they've got something protective going on. So I'd say right now, yeah, don't know what we can do about that. Again, as we learn more about the biology, hopefully we can start targeting medicines or other types of treatments, not necessarily medications to help. But at this point, yeah, it's more do no harm. And like I said, we're looking at that. We're doing a teens study right now where we've got an intervention with teens that are overweight, and we're doing a follow-up 2, 3, 4 years down the line to see if the intervention that we're doing is in fact helping them maintain their lower weights as well as not sending them into an area of potential psychological risk, right? So results on that aren't out yet.
Yo agregaría eso que preguntaron, que no se pierdan la siguiente sesión de anorexia nervosa atípica, donde vamos a hablar un poco de esta supresión de peso y del gap nutricio, y que sucede mucho en los pacientes que tienen sobrepeso. Another question, Laura, is seeing the rise of subclinical eating disorders and bad relationships with food, Do you think genetics played a role too in these issues? Is there any research on that topic?
Can you repeat that?
Sure.
So I understand the last part. Is there any research on it? But yeah.
It's saying the rise of subclinical eating disorders and bad
yes.
relationship with food. Do you think genetics played a role too in these issues?
I think absolutely genetics can play a role in those issues. You know, the sub-threshold eating disorders have always been there. I think part of the rise that you might be seeing too is more attention. It's more, people are much more aware of issues now, of eating disorders and everything else. So it's come sort of out of the closet and into the open, and people are more willing to talk about it. That could be part of the rise that you're seeing. If it's not, if it's truly that there is this rise, it absolutely can have a genetic component. It could also be environment. I mean, right now there's a lot of triggers in this world for people. We know that COVID has really impacted the eating disorders potentially negatively across the globe.
Okay, is there a role for the microbiome in eating disorders? And also, how can we avoid the predisposition when we explain to the patient like, oh, you have the anorexia gene or the eating disorders gene?
Okay, so microbiome, absolutely. We do a lot of research on the microbiome, and we are moving forward in a lot of ways of that, because we know that the gut talks to the brain and the brain talks to the gut. And there's a lot of animal models research being done on anorexia or eating disorders or heavy obesity stuff and the microbiome. We're just beginning to understand all of that. And again, as for the gene for anorexia, again, it's not one gene. It is many, 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 many genes. And we've only identified a very small subset of genes. And again, as I was saying, just because you may have a really high genetic predisposition for anorexia, you may also have protective factors. So you will never develop the disorder. And the same thing with, you may have a very low risk, but something is triggering. So yeah, you really can't talk to patients about what their genetic risk is at this time. There's just no way to go ahead and say there is no gene, it's many genes. And the only way we know that you actually have a high risk right now without actually going in and doing DNA stuff is look at family history. And then, yeah, you might have a slight increase. But as I showed in the one graph where they were looking at the research, that people with eating disorders tend to seriously overestimate the risk that their child will develop an eating disorder. They were 8 times higher. They thought their child's risk was 8 times higher than what it actually is. So I hope that answered the question.
Yes, perfect.
Okay.
Do you think there's going to be a significant difference between the correlations that were found and the new correlations because of the inclusion of more ethnicities?
So we will actually look at these by ethnicity because the genetic background is going to be very different. So we will do folks that have that are of European ancestry. We'll do those of African ancestry. We'll do those of we'll separate the Latinas out and do those because we don't know. We don't know what's going to pop out for each one. We can include them all then in a meta-analysis after you take them apart to see what is common across all of them. But I imagine that some of the profiles are going to look a little bit different. And that means that some of the genetic correlations will look a little bit different.
Muy bien, pues ya estamos sobre el tiempo, entonces tenemos que cerrar. Muchas gracias a todos. Les quiero comentar que los próximos 3 miércoles de octubre tendremos otras sesiones de trastornos de la conducta alimentaria. La siguiente sesión es sobre anorexia nervosa atípica. Después de esa tenemos la neurobiología de los trastornos de la conducta alimentaria y finalmente trastornos de la conducta alimentaria en niños, todos por mentores y excelentes profesionales en el campo de los trastornos de la conducta alimentaria, así que aquí los esperamos. Laura, thank you so much for your presence here. We are very grateful about having you here, and we'll continue in touch. Thank you so much.
Thank you. Thank you. Have a great day.
Thank you.
Thank you.
Bye.
Thank you so much, Dr. Laura and Dr. Eva. Have a good day. Muchas gracias a toda nuestra audiencia y los esperamos la próxima semana. Les recordamos también nos hagan favor de llenar la encuesta de satisfacción. La liga está en el chat.